analytical techniques. So you guys can see the slide, right? Okay. So basically here we will focus on the general steps or general guideline on how does a method is being developed. It means that if you have a sample, if you are given a sample, and then people ask you, please analyze this sample. So first you have to know the identity of the sample, what kind of test you want to do. So once you determine and what kind of parameter that you want to measure, for example, and once you have determined all of that, so you must have a method to do the testing or to do the analysis. So this is where all of this general guideline will comes in. Okay, so you cannot just randomly create a method. So simply just, oh, let's just use this kind of method to do, for example, HPLC testing. So I just add a little bit of this and use any kind of solvent. So there must be a certain guide that you have to follow. So some of the content will be on, the first part will be on the method development itself, and then later we will see what is the types of biological sample, matrix effect, sample recovery, signal to noise ratio, and also standards, okay, which is divided into internal and external. So some of keywords or terms that you have to familiarize yourself in the line of pharmaceutical analysis is, one is analyte. So analyte is the component of a system to be analyzed. So any part of that you want to analyze, for example, you have a tablet of what do you call that, paracetamol, for example, and you want to analyze the content of paracetamol API inside each tablet. So the PCM itself is the analyte, okay, not the other, for example, binders inside, the coloring. So that is not considered to be analyte. And then an analysis is the process of breaking complex or substance into smaller parts in order to gain a better understanding of it. And method is the way for you to achieve the parameters that you want to measure. So a systematic procedure or a technique of doing something or way doing something. Okay, especially it must be in accordance with a definite plan. So you must have a plan first. This is very important. So you plan ahead, you already know what to do. First step is to do this, next step to do, let's say, first step you weigh and then you dissolve, then only you start with the analysis. So you must follow this plan and sequence. So the definition of test method according to ASTM. ASTM is American Society of Testing and Measurement. Okay, it is a body in the United States where they came out with a guideline of how to do testing and measurements. Okay. By their definition, a test method is any definitive procedure to identify, to measure, or to evaluate a material, product, system, or service that produces result. So at the end of the testing, you must always obtain a result, whether the result can be used or not, whether it passed the specification or not, that is another matter. Okay, as long as the test method itself must obtain a result. So methods should include, so these are the things that you have to take into account in terms of sampling. So sampling if you have 1,000 tablets, so how much of samples do you need to represent those 1,000 tablets? So you cannot just take one. Probably you need to do random sampling. Okay. Then handling of the sample. If the sample is volatile or probably light sensitive, so that have to be determined earlier. And then in terms of transportation and storage, where it should be kept and where it should be do, and the sample preparation itself. So before you start doing this testing, the sample must be prepped. Sometimes samples came in the form of solid, but the testing method itself or the instrument that you are using, for example HPLC, sample must be in liquid form. Okay, so you must have preparation. The sample must be prepared first. So you have to weigh, and then you have to dissolve into solution phase. Then only it can be used to be analyzed. Okay, so the lab must always have instruction on the use and operation of all relevant equipment. So this is one of the requirements for ISO 17025. So ISO 17025 is related with lab. Okay, so if you have or if you see any lab that has the certification of what ISO 17025, then that lab must always in good condition. Okay, it follows it has records. If you want to enter, you must wear all the PPEs. Okay, and they do calibration according to schedule. Okay, this is how to say, a good lab will thrive to get this ISO certification. Okay, normally you see in office we have this ISO 9001, 9002. Okay, and that one is related to office management. Okay, but for lab, it has a very specific ISO for itself, which is ISO 17025. And normally, not many lab in pharmaceutical manufacturing company in Malaysia has this certification. I know in my old previous company in Pharmaniaga, only one of their lab is certified 17025, which is in the one in Shah Alam. Somebody joining in, let me let them to join. Okay. So minimum method information. So these are some of the checklists that you have to have before starting with your method validation. In terms of first, we have that appropriate identification. So what kind of measurement you want to do, whether it is in terms of weight or volume or concentration, and then scope, description of the type of item to be tested or calibrated. So information of the sample itself. Okay, if you want to test, for example, raw material. Okay, if you end up in QC and then you receive raw material sample to be used in the manufacturing of drug, so those description of the sample must be available first, and then parameters and or quantities to be determined. And then we have this equipment. So what kind of equipment that you want to use? GC or IR, or in fact even UV or pH meter. So that information must be prepared first. So that if let's say you want to do pH testing, so end up once you prepare all the sample, then you see that, oh, the pH meter is broken, cannot be used, it's not calibrated. So it's kind of a waste of time and also waste of your chemical during the sample prep. So environmental conditions, okay, description of the procedure. So this is the procedure of the testing itself. Should include affixing identification marks. So if you do your testing, normally test sample will be in volumetric flask or could be in a beaker. So it must be labeled so that you won't get confused. Or if let's say another person comes in into the lab and probably you were not around for a while, okay, you go to the other side of the room to take solvent or anything, they the other guy may not throw it or contaminate it, so that it's important to label. Somebody is joining. Yes. Okay. Wait. And then you have to check on the equipment properly calibrated and adjusted. So method of recording the observation and result. So this one, normally most of the instrument that we have nowadays, they will record automatically because most of them are connected to computers. But some still have to be manually taken. I mean the result. Okay, for example, like what do you call that? Weighing balance. So some of it are not connected to printers. So you have to manually taken down what is the weight. So it may appear on the display, but there's no way of recording that value. Okay. So safety measures, and then the last three will be criteria or requirement for approval or rejection. And this one is with regards to if you get the result already, so whether the product is accepted or not. So that is based on other specification. So standard method. So this is important. This is different than standard. Okay, this is standard methods. Normally you can obtain through what you call this reputable organization or entity. Okay, so this is a method that has already been proven. So what you can do, if you let's say a sample, if you have a sample and you don't want to develop the method from scratch, so you can refer to this. For example, we have ASTM, ISO organization, JPCS, this one EPA, United States Environmental Protection Agency, or especially for pharmaceutical products, you have to refer to pharmacopoeia. Okay, you have USP, United States Pharmacopoeia, British Pharmacopoeia. These two are the most referred pharmacopoeia, especially in our Malaysian market. Okay, most manufacturing company will try to refer to these two, but they are not free lah, so you have to pay for subscription. Another pharmacopoeia is if let's say these two, if you have any samples and the methods are not listed in any of these two, you can try to also refer in European pharmacopoeia and also Japanese pharmacopoeia. The Japanese pharmacopoeia is free, so you can even now you can just Google Japanese pharmacopoeia and you can get all the test method that is available. Okay, and Chinese pharmacopoeia is also very they have a lot of different kinds of material in terms of test method, but not really used as a guide due to the reputability. But then again, if it's not available in other pharmacopoeia, then that can be your last choice as well. Okay, so standard methods is any appropriate methods that have been published, okay, either in international, regional, or national standards, or by a reputable technical organization, or in fact, in relevant scientific texts or journals. So scientific texts or journals is in article published in journals. That means by other researchers. Okay, so this will be very helpful when you do your final year project. If let's say your final year project will involve lab what you call lab testing, okay, either pharmacology or pharmaceutic or in fact even chemistry, pharmaceutical chemistry. So you have to refer to articles in journals, okay, for methods to analyze a sample or to synthesize a chemical compound, or in fact to do what you call that in synthesis, to formulate a drug. So this is where you can try to refer the methods, and these are called as standard methods because it is already being reported and they obtain result with that. Another way is to develop a method is non-standard method. So non-standard meaning you do not follow any of that, okay, but the method is new, so and not covered by any of the standard methods. New meaning you yourself can create your own methods. So the method should be developed and validated appropriately before use. Let's say you already try to copy the method from USP, United States Pharmacopoeia, but hey, you have tried for many times, probably you have repeated up to 5 or 6 times, you still did not get the intended result. Okay, or another reason is all of the methods that is in the standard method you cannot do in your lab. Probably you don't have the equipment or you don't have the chemical, so you have to try to improvise and develop a new method which suit yourself and also the lab that you are in. So this is called as non-standard. But all of this new method must be validated. Validated means you have to confirm that that method can be used and can be repeated. Repeated in a way that you do it today, you get a result. You do it tomorrow, you get the same result. This is, let's say, if you use the same sample, you get the same result. If you do it next week also, you still get the same result. And if let's say another person is doing the test or the analysis, they should obtain the same result as well. So that is what is meant by validated. And for validation of method validation, there are several parameters that has to be fulfilled. This one will be covered in the next chapter by Ms. Yasmin. Okay. So in valid method, so in validation, like I said just now, is a confirmation by examination and the provision of objective evidence that the particular requirements for a specific intended use are fulfilled. This is sort of like a proper definition, okay? But you just want to validate. Okay, the laboratory shall validate. So these are the things that need validation. Non-standard methods, okay? Laboratory design or develop method. So this one, you develop the method based on your lab feasibility. Standard methods used outside their intended scope. Let's say you found standard methods, but you want to use that method for another sample, another product. Let's say in the standard methods, that testing should be only for glycoside medication, okay, or probably for antibiotic for cefuroxime, for example. But then you want to do that testing for amoxicillin, for example, another type of antibiotic. So that has to be validated as well, okay? And modification of standard method. So if you modify standard method obtained from the references, so this one also you have to do validation. So normally this is what we call as partial validation. You only validate the parts that you modify. So method development is a process that determines test method capabilities through selection and optimization of analytical test parameters. Okay, this is the definition of method development. Okay, it will provide assurance that the test method that you are using will meet the requirement, or in fact even surpass. So let's say it is even better, okay, than the old method, for example. Okay, the method developed shall have been validated appropriately before use. So if your reference is standard method, then you do not have to be validated. But if it's under all of the requirement like this one, okay, all of this, so this you have to validate first. So as method development proceeds, regular review should be carried out. That means you should see after each steps of the analysis, is there any problem, is there any error that keep on arising? So you have to take note of that. So the importance of method development, one is for improvement in terms of testing performance. Okay, by employing method development principles to standard test methods. Okay, this is when you apply to new products or new material. So during method development process, the optimum test parameters are identified to ensure applicability and reliability of the data. Okay, so before we go into the plan, so normally the use of this method development, if you work later on, if you work in pharmaceutical manufacturing companies, okay, the method development will normally be employed in R&D, in research and development, because this is where if you have any new products that you want to sell, you must test the product first, right? The new product. So this is where person in charge will develop a new method for testing. Once everything is validated, this new method, it will just be transferred to the quality control. Okay, quality control normally will just have to do modification, but not full method development. So this what you call method development will take times. Okay, sometimes it will be like, if you're lucky, then in one day you can obtain the result straight away. But it will take, in fact, weeks, 1 or 2 weeks, to have a complete set of method. That is not included in validation. For validation, you have to add another 2 to 3 weeks. Okay, so for pharmaceutical products, normally the test will involve using HPLC, and HPLC is a very sensitive how to say, instrument. Okay, if you change a little bits of the parameter, for example, flow rate of the solvent or types of solvent used, then you will end up with different kinds of result altogether. So and you have to remember that all your tests must follow specification. So there are certain things that that is allowed. Okay, for example, in HPLC, the result will be in chromatogram. It looks like a graph. Okay, so the size of the peak should be there's a limit. Okay, in terms of size that you have to achieve. So if it's not up to the standards, then you have to start all over again. So it will take some time to develop method for new products. Okay, for new products. So now we will see the method development plan. Okay, so I think there are 6 of them. Okay, this one. So before we go into that, so this is ISO 17025, which is to certify lab according to the ISO standard. Okay, so in ISO 17025, in terms of selection of method, laboratory developed methods or methods adopted may also be used if they are appropriate for the intended use and if they are validated. So this is according to ISO 17025. So if you want to use method other than the standard methods, then you have to validate it first. Okay, so the introduction of test and calibration methods developed by the lab shall be a planned activity. See, very important, a planned activity. So you must follow the step accordingly. So you cannot jump. Or let's say you start step number 1 and then suddenly you want to go to step 6. So you must follow step 1, step 2, step 3 and so on. Okay. And it shall be assigned to qualified personnel equipped with adequate resources. So in a lab with ISO 17025, even though you go in, you have a degree or in fact you have a PhD, but you must be, how to say, it's not like the word tested. So you have to conduct sort of like an exam lah. Okay, you have to test a sample, you have to analyze a sample, and it should be verified by your superior, normally a manager or something, that the result is good to go. It is acceptable. Then only you can do testing for all the other products. If you do not do this, and this one must be done every year. So every year you have to sort of like calibrate yourself. Okay, all of the personnel must be calibrated itself, or in a way. So you have to do a testing. Everybody will do the same testing, okay, and you should get the same result, all of you, so that we can say that you are a qualified personnel. Then only, this is one of the requirement for ISO 17125. Okay, so the method development plan involve, the first one is studying the sample. Okay, then establish your goals or your objective, method objective. The third one will be literature review. Then the selection of the method you want to choose. Okay, once you have several types of methods involved, then you must choose the best one for you that suits yourself and the lab that you are in. And number 5 will be to optimize the method. So you want to make sure that the method is the best it can be, and it can be used for a long time. And anybody that use that method will get the results. Okay. And the last one is to finalize the method. First, what is studying the sample means? So if you have a sample, you must have all the available data about the sample. It means by what is the composition. So this can correspond to the, for example, chemical formula, or whether it contain hazardous, whether the sample is hazardous material or not, whether it is flammable, okay, poisonous. So this one is one of the important criteria or parameters of the samples that you have to know. And then sample handling and storage. So how the sample should be handled. Okay, like I said earlier, whether some is light sensitive, you have to put in amber bottle. You cannot use normal clear glass. Okay, you have to put in an amber bottle. And storage, whether it needs to be in below 0 degrees Celsius, or you can just put in a normal fridge, or just at room temperature. So what is the physical and chemical properties and reaction with the environment? This one, if let's say the chemical is highly, what do you call that? Easy to absorb moisture. So you have to put it in a dry room, okay, with a humidifier, so that there's no moisture that can be absorbed by the sample. And then responding with the used equipments and any other properties that is relevant to your testing. Next is your goals or your objective. So why do you want to develop this method? So what kind of parameters that you want to measure? So this is based okay, all of the goals will be met at the end of the method development process. So these are some of the example of your objectives. So if you want to detect something, so it is a qualitative identification. Okay, if you want to quantitate, then quantitative determination. Purity, if you want to check the purity of a sample, okay, This an impurity may be present which interferes with quantifying the component of interest. And then characterize what are the compound properties, or in fact in characterization you can see what is the compound made out of. Okay, what kind of functional group that it has. Probably it contain hydroxy group OH or an amino group, things like that. And then to purify, collect the compound for other use. This one, you have a sample and then you want to purify it so that it can be collected. So you want to remove any impurities. Okay. Then you have to conduct your literature review. Literature review, it means based on the first few slides, like all of those standard methods. Okay, you can refer any previously developed methods, okay, with quantitation and sample matrices that are close to your requirement and can form a starting point for your method. So sometimes, if you want to find a method, you may not find anything that fully suit you 100%. Sometimes you have to take 3 methods and then combine them together so that it can be used to do your kind of testing. So resources in which you can search. Okay, now everybody have internet, so that is the first thing that they do. Okay, just go to Google, type testing for what material. So from there also, we can also refer to pharmacopoeia. So USP, BP, or Japanese pharmacopoeia or European pharmacopoeia. And then in the US, or if you want to sell your product to the US, then you must also follow FDA requirement. FDA is Food and Drugs Agency in the US. Okay, if you just want to sell in Malaysia, then you have to follow our guideline like NPRA. Okay, and then EPA requirement, USDA methods. Okay, colleagues, you can ask your friend as well, and professional technical journals and setting, or in fact corporate application notes. Okay, so these are some of the references that you can use to find the method that is suitable for your use. Next is select an approach. So from here, okay, don't just take one only. Okay, have a few methods that you can later on see in detail before choosing. So here in step 4, choose a standard test method that is appropriate for your analysis on similar materials. Okay, it is good to begin method development with a standard test method and make slight modification or improvement as needed. So once when you do your literature review, don't straight away say that, oh, I just developed a new method, new non-standard method. Instead, try to have a standard method as your base, then only you make slight adjustment to it. Okay. So if a standard test method is not available, then only it is necessary to develop new methods. But this is always try to avoid this. Okay, normally we will try our best to have a standard test method first before venturing into developing a new test method, because this requires full validation of your test method. Okay. Then the next step number 5, is to optimize the method. Okay, critical test parameters that need to be identified and evaluated by your analysis, designed to determine a suitable operating range for each one. Or critical test parameters were defined as the operating factors that when varied, so varied meaning you make slight adjustment, significantly affect the result. So you try to see which part of your analysis, if you change a little bit, it will give a very different result For example, in the test method, it said that measure or mix your sample with 10 ml of sodium hydroxide, probably to dissolve it. What happens if, let's say, you add more than 10 ml? So you add 11 ml, for example, or you the sodium hydroxide is added less than 10 ml, 9 ml. So you have to check these two values. If it's affect the result that you obtain greatly, then you should know that this part is very crucial. So make sure that it has to be followed accordingly as stated. But if, let's say, whether the volume of that sodium hydroxide can be, if you add less or more, it doesn't affect the result, then in your method, we know that that part is not really that critical. So you can just say that add about 10 ml. If not, you have to exactly say add 10, exactly 10 ml of sodium hydroxide. Okay. So this is what it means by optimizing the method. So you see which part of the method that have great effect on the result that you will obtain later. And method development activity should be documented. So anything or any adjustment you made, make sure it is documented so that you know what kind of adjustment that you have made. Otherwise, if you do not make this documentation, sometimes you may end up back to the original standard method. For example, original standard method is addition of 5 ml of HCl to the sample mixture, and then you try to change to 4 or maybe 3. But if you don't document it, probably you're going to end up back to number 5. Let's say, oh, I haven't done 5 ml. So you do back, which is the origin. So try to have a documentation. This is example of standard HPLC test parameters. So this is HPLC. In our lab, we have two types. In the analysis lab, we have the semi-prep and also analytical HPLC. The semi-prep is one that can collect your sample afterwards. After you've done your analysis, it can collect back your sample. But the analysis is a destructive method in which your sample use is very small, but then again, you cannot collect it back. So some of the test parameters for HPLC, as you can see, there's a lot to be take into account. So many things that you can to change. For example, columns. So HPLC column, the size of a normal HPLC column is like the size of a pen or your pencil. So that is very small. But the each of the column has different packing. Okay, as you can see, this is the brand is Thermoscientific Accuro, and this one, the brand is Thermoscientific but different model, Synchronis. And as you can see, this is 2.6 micrometer. This one is 1.7 micrometer. This is actually the size of the silica inside the column itself. See different sizes, and this one is the length of the column, 2.1 times 10 mm, 10 cm. Okay, so different types of column. And then eluents. Eluents is the mobile phase or the solvent that you want to use to move the sample throughout this whole system. Okay, this one on top of the machine, this is the eluents or what we call as mobile phase. Okay, some system can have up to 4 different mobile phase that can be pumped into the system, A, B, C, D. In our lab, our HPLC can pump up to 2 types of mobile phase. Okay, so the more pump the HPLC have, more expensive. And so this one, for example, defined by solvent selection valve. Okay, you can have water with a little bit of phosphoric acid, formic acid. Okay, and then you have acetonitrile, methanol, and water acetonitrile mixtures. Then another thing that you have to consider is this A, B, C, D can be mixed together. Let's say you want to mix between acetonitrile and methanol. So this HPLC can do that. So you don't have to pre-mix. Okay, you can mix, you can set in the computer, I want acetonitrile 50%, methanol 50%, then it will inject. Or you want acetonitrile 60%, methanol 40%. So these are some of the parameters that you can try to change. Okay, and even if you change by 1%, then you are gonna see different result. And this one is gradient. Gradient is the combination of the mobile phase. Okay, this one next semester in pharmaceutical analysis 2, you will study more on HPLC. Okay, flow rate. Flow rate is how fast all of this solvent is being pumped into this whole system. Okay, this one is 0.8, or it can be 0.4, or probably you want to pump it slightly faster, then you can increase to 1.0 or 1.5 or 2. Okay, then pressure. Okay, pressure inside the system from the pump itself, and temperature also you can change 40 degrees, 35 degrees, 37, 50 degrees, okay, depending on the sample. And injection. Injection is the amount of sample to be tested. So once you have done your sample preparation, this is the amount that will be used to be tested inside this HPLC. Very, very small, just 1 microliter. Okay, or sometimes they use 1.5. Okay, and the detection, okay, detector, normally this one, which is a UV. So what kind of wavelength that you want to use? So normally the wavelength is obtained from your literature earlier, which can be obtained from pharmacopoeia, for example. For each specific compound, for example, like caffeine, they have specific wavelength that can be used to detect the presence of caffeine, for example. Okay, these are some of the adjustment ranges. So the allowed changes. Okay, this one you can read. For example, like column length, what is the allowed changes can be made up to 70%. It can be more or less. So for example, this one, the length is 10 cm. So you can increase 70%, which is 17 cm, or decrease 10 minus 7, 3 cm. So that is it. So you cannot change more than that. Then the diameter, UV wavelength. So this one UV wavelength, no changes outside manufacturer specification. So you cannot change in terms of UV wavelength. And the last thing to do is finalize your method. So once everything is settled, you're happy with the with all the results, okay, all the equipments are there, all materials can available in your lab, so you finalize your method. So final step in the method development process is to evaluate actual samples by the new or improved standard method. Okay, evaluate samples at the upper and lower limit. Comparison of result generated using control samples. So you must have control samples. And documented test method should be revised to include any changes made to the procedure, okay, during finalization step, or if you want to add any other information, additional information. Okay, this is required to formally validate the newly developed or improved standard test method. So after finalize the method, okay, if let's say your method is not a standard method or a slightly modified standard method, all of this information is needed so that you can do your validation. Types of sample. So this here is the vial for your HPLC. So the vial is very small. It's like what do you call that? Satu ruas jari, your fingers. Okay, the length is very small. So that's why I think here you can put up to 10 microliter of solution. Okay, normally needle will come from top and inject into the bottle. Okay, then only it will be tested or analyzed in the HPLC machine. So type of sample that you need to consider or the parameters of the sample that you need to consider is state. What is the state at room temperature? Okay, whether it is solid or liquid, and if the sample is organic or inorganic, or even if it is a biological sample, and solubility in water and organic solvent, melting boiling point. These are the physical and chemical properties. Okay. Continuation. You have pH stability, chirality, reaction towards heating, fluorescence, and so on. Example of biological samples. So just now everything is in the product itself, but once you have a product and if you want to register a new product with what you call government agency, for example, Malaysia, the NPRA, okay, National Pharmaceutical Regulatory Agency, you have to conduct a clinical test for the drug, the new drug. And this clinical test will involve giving human, okay, no human test sample, the drug, okay. And what they do is, let's say the drug is supposed to be in your body for 6 hours. Once the patient consume the drugs, after half an hour, they will draw blood. They will take some blood from the patients, okay, normally at an interval, probably every 1 hour or 2 hours. And this drug will be tested to see the content of what you call your API just now. Okay, let's say it is a drug to treat hypertension, for example. Then we will see what is the amount of drug in that blood. So an example of biological samples. First, we have like cheek tissue. This one is to check for DNA profiling, and then dried blood spot. You can check your DNA with this or RNA. Okay, using what do you call that? PCR method. Like for example, now they want to check for COVID virus. So that is also an example of biological sample because it is coming from a living thing, okay, living human being, okay, in which they will check throat sample. They will take swab of your throat and your nose and see if they contain any COVID-19 virus or not. That is using the standard PCR method lah. Okay, and organ tissue, this one is normally what you call as biopsy, normally done to check for cancer, things like that. Plasma also considered biological sample. Urine, urine test. This is also most of you have done urine test. Okay, feces or stool sample, and then skin, and last one is hair. Okay, this is part of biological sample. Okay, this from now on until the rest of the lectures is some of the terms that is commonly used in analysis or that you will encounter in method development process, so that you know what to avoid and so on. So for example, like the first one is matrix effect. Okay, matrix refers to the components of a sample other than the analyte of interest. So any other things beside the analyte, we consider it to be matrix. Okay, and the matrix effect is it can have it will have a considerable effect on the way the analysis is conducted and the quality in which your result will be obtained. Such effect is called matrix. So if too much of the other parts of the sample beside the analyte, we want to see whether it will affect your result or not. So if it does affect the result, then this is called something like matrix effects. Okay. So it can be ruled out by using blank. So this is what is mostly practiced. Okay, blank is normally if you want to remove the consideration for the solvent. Okay, let's say you conduct UV testing. I believe most of you have used UV before. Okay, to do UV testing, if the sample is solid, you need to dissolve it in certain kind of solvent. Let's say water. For example, if your sample is potassium permanganate, so you dissolve that potassium permanganate in water, then you measure. But then again, you are measuring the water and also potassium permanganate. So how are you going to remove the effect of the water, or to calculate how much of value for that water alone? So you use this blank. So the blank is normally any solvent that you use to dissolve the sample itself. Okay, in UV spectroscopy, when a particular solution is made of solvent X, just before each sample is being analyzed, a blank reading usually only solvent X is taken. Then only you can start with the measurement of the samples itself. Sample recovery. So this is in a way that once you do your analysis, do you still want your sample or not? So one of the criteria in which you have to consider when you do your method development. Okay, in terms of, is it necessary for the sample to be recovered after analysis? So many methods of analysis are destructive. Okay, and even if the method is not destructive, most of the detectors are destructive. So in many scenario, the sample to be analyzed will only be available in small amount. Okay, in this case, the sample has to be retrieved after analysis for further different types of analysis. So if let's say your sample is very small, small not in terms of size, small in terms of the amount, okay, and you only have enough or sufficient to do a few testing, okay, and let's say the test that you done need to be repeated again. So you have to consider this, meaning that once you do your uh, test, you want to collect back the sample. Okay, you have, so in a way you have to find a method in which that your sample can be collected. For example, in IR spectroscopy, okay, the usual method will involve mixing your sample with potassium bromide, KBr, okay, and then only put into the machine. In a way, the sample is not really being destroyed, but once you mix the sample with potassium bromide, then it is very hard to separate, because the sample will be in solid. So let's say you have solid A and then you have solid B. Then you mix them together and then you grind. You have to grind everything together into powder. So how do you want to separate these two powder? So it's very hard. The solution for IR is there is another type of or attachment that you can use so that you can straight away put the IR itself onto the machine, and this is what the attachment is called ATR, attenuated total reflectance. Okay, and it includes one very small tiny diamond on the accessory itself to the lab. In our lab, we just have the normal KBR method because the accessory for the ATR, the price is, if I'm not mistaken, depending on the type of the IR, could be around $50,000. Okay, you can buy a car with that thing. And if you're using that method, then the sample can be collected back, no problem. And another type of, for example, analysis that you can collect back is HPLC. In our lab, we have both type. The semi-prep HPLC, you have a collector. So after it is run through the UV detector, then your sample can be collected back. But the other HPLC, which is the analytical HPLC, That one, after you done your analysis, it will straight go into the waste bottle. Okay, so this is one of the criteria that you have to consider when you want to develop your method. So this one, you have destructive and non-destructive method. So destructive methods, the first one is most chromatography techniques. Okay, but with exception, okay, with exception, the semi-prep HPLC and IR. If you are using the ATR method, then that is not destructive. Okay, if you use other methods like the KBR or Nujol, this one next semester you will learn what is IR. So that is destructive. Non-destructive method like NMR. NMR you can take back. NMR stands for nuclear magnetic resonance. Fluorescence. UV, it is not destructive, but then again, if your sample is in solid, you already dissolve it in solvent. So in a way, you can collect back, but then you have to figure out how to remove the solvent. Okay, but if your sample is already in liquid form, then no problem. Another term that you have to know is signal to noise ratio. This will mainly be seen for instrument that have what you call result like this, okay, in a graph form. For example, in UV, in double beam UV spectrophotometer, the result will come out in a graph, not in just numbers. And this is example for HPLC or GC, gas chromatography, or high performance liquid chromatography. And this is called as a chromatogram. So what does it mean by signal to noise ratio? Noise is this one, all this jagged peak, very small peak at the bottom. Okay, this is considered to be noise. This is not the thing you want to measure. Signal is the result itself, the peaks that is in the peaks of interest. Let's say this is the peak of your sample. So this is the signal. And signal to noise ratio is normally measured by height of signal and then noise in terms of RMS value. So this can be done if you are using HPLC, normally connected to a computer. The software inside it will calculate. So it will give a value and there is amount that you have to follow lah. If it's exceed the amount for the signal to noise ratio, then you have to make adjustment to the parameters of the HPLC. Okay, so the ratio of the strength of an electrical or other signal carrying information to that of unwanted interference. So how much noise is being produced when that particular substance is tested with the chosen technique. And if too much noise, this one, if too much noise, then your method is not suitable. So you have to make adjustment. The larger the signal to noise ratio, the better the method is. So value this one, larger, meaning you have very good signal and very, very small noise. Okay, that is better. And then standards. Okay, standards. So when you want to do a testing, let's say for caffeine, you want to detect caffeine content in Coke, for example. So you take a sample of the Coke drink and then you put it into the HPLC machine. Then you're going to get peaks like this. It will come out as peak like this. And this is time, probably 5 minutes, 10 minutes, and so on. But this peak is not labeled. The machine does not know this peak is caffeine or any other stuff. And if the sample contain more than one peak, for example, you have one here and then another one very big over here, then how are you going to know which of the peak is caffeine? So this can be achieved by using standard. So you have to purchase standard of caffeine. This can be purchased through what do you call that, chemical suppliers okay, they do sell standards for various chemical. Okay, you have to do purchase this standard of caffeine, for example, and then prepare a standard solution. Okay, this standard solution containing a precisely known concentration of an element or a substance. Okay, then you take this and analyze it again. Analyze it also together with the sample, and then only you can compare between these two. Oh, you know that, oh, at 5 minutes, for example, at 5 minutes, you have this peak. And for your sample, the peaks also is at 5 minutes. So then you know that, oh, this is the peak for caffeine, for example. So it can also be used as control. And in terms of standards, there are primary and secondary standards, and also internal and external standards. So this is the pictogram of how standard solutions are normally being prepared. First, you weigh, okay, and it must always be prepared in a volumetric flask. Okay, you don't use beaker anymore. In pharmaceutical analysis, you don't prepare the solution in a beaker. A beaker is not accurate. Okay, prepare using this volumetric flask. Okay, this is called as a volumetric flask. And you don't measure using measuring cylinder anymore. We use pipette. Okay, pipette is more accurate than measuring cylinder. Okay, so once you weigh, put it into this, and then top up with the solvent until this mark. Okay, then only you can obtain your standard solution. Okay, let's say this one is, I think, 500 ml. So you don't use 500 ml beaker. You take you take a beaker. You measure 500 ml solution using measuring cylinder. No, that is not how you do it in analysis. So a primary standard is a substance of known high purity, which may be dissolved in a known volume of solvent to give a primary standard solution with accurately known concentration. Okay, and a secondary standard that is prepared in the laboratory for specific analysis, usually standardized against a primary standard. So, in a pharmaceutical manufacturing environment, this primary standard is the standard that is sold, or you can buy it from pharmacopoeia company like USP or BP. They do sell primary standard of various active pharmaceutical ingredient, and the price is not cheap. Okay, for example, 10 mg of certain API, 10 milligram, not gram, milligram, can cost you about upwards to 2,000 USD dollars for 10 for about 10 milligram mg, very very small amount, and you can only use that for 1 or 2 tests only. So every testing you do, you have to include a standard. So if let's say you want to do 10 tests, 10 different kind of tests that need standard, and you only have each bottle or each vial of primary standard is only like 10 milligram. So it will cost a lot of money. So instead of that, you need a secondary standard. So a secondary standard is normally the raw material that you obtain, which is cheaper in price. Okay, so those raw material will be standardized against this primary standard. Then only it can be used as a secondary. But this one has been done, there's an expiry date lah. So normally it is done every 3 months, the standardization of this secondary standard. Okay, normally it is done every 3 months or every half a year, 6 months. Okay. Then in terms of internal standard, it is a chemical substance that is added in constant amount to samples. Okay, the blank and calibration standards in chemical analysis. So this is done to correct for the loss of analyte during the analysis. So that means the internal standard is added to the sample mixture itself. Okay, you don't separate it and analyze it separately. So for instance, adding a fixed amount of standard into a negative control, positive control in every sample analysis. So any reading obtained then shows deviation, and the amount standard added will apply to the sample as well. Okay, so it is also used when analytical technique use has not been properly validated. So the one below, so one thing that you have to remember before using that standard, any standard to be used for analysis, that means you want to compare it to a sample, you have to do a calibration study or calibration standard or calibration curve. Okay, please remember calibration curve like this. So what you have to do is you have to prepare that standard at different concentration. You see, for example, like this one, you have this one most concentrated, and then slightly less, less, and the last one, this one has no standard in it at all. Then what you do is you measure with whatever kind of analysis that you do, whether it is HPLC or UV, for example, and then you have to plot a graph between the concentration and the value of the signal. Okay, whether it is absorbance or concentration, or okay. So the result obtained should be in a straight line like this. Okay, straight line, and the R squared. So from Excel, you can use the what do you call that? Show the equation and then show the R squared value, the intercept. So this R squared for a pharmaceutical analysis, the acceptable value is 0.99xx. So that means you can start from 0.9901 until the maximum is 1.0. Okay, so that is the best is to get 1.0, but very hardly. So if you can get 0.9975, for example, that is good enough. Then only the standard can be used to together with the sample. Okay, and the last one is external external standard, like internal standard, okay, with known behavior, but it is not added to the unknown. Okay, sample preparation or sample inlet, rather it is run alone. So this one is like normally, so you have a standard, you prepare, and then you run the analysis just by itself, okay, as a sample, and usually at different concentration, so you can generate a standard curve, which is this one. Okay, so you can generate standard curve. So the standard curve will be used as reference for all analysis that is done afterwards. So let's say you have a sample with unknown concentration, so you measure, and then it will have a peak at, let's say, 35,000. So you can just extrapolate from here until here, go down. So you know what is the concentration. Okay, so that is all for this topic. So if you have any